I've been working on this project for over a year now, around a year now. My project is about determining the effects that Alzheimer's disease risk factors have on the functional networks of mouse models. Some primary motivations of our study are based on the fact that Alzheimer's disease pathology can be seen in patients years before symptoms even appear. The risk factors that we were interested in examining were APOE genotype, sex, age, and obesity. There are three major APOE allelic variants. There's APOE2, which confers neuroprotection and is found in around 5 to 10% of the US population. There's also APOE3, which is a control allele, and it has a neutral effect that's found around 75% of the US population. And lastly, as you've probably heard before from previous presentations, APOE4 is a high-risk allele that's found in around 50% of Alzheimer's disease patients. Mouse models are often used in Alzheimer's disease studies because they result in more controlled studies and they age a lot faster. We use mice to model genetic risk for Alzheimer's disease, and we replicate the human APOE allele variants, APOE2, APOE3, and APOE4. We also investigate the impact that sex, age, diet, and genotype can have on the functional networks of mouse models. We had two main research questions. We examined local and global properties of connectomes. Connectomes are connectivity matrices, and I'll explain that more later on. We looked at how specific regions can exhibit significant functional activity. We used 346 mouse models for our study. The majority of our mice belonged to the APOE2 genotype. We had around 110 mice for the high-fat diet group, and that meant they were on a diet with around 40% fat. Our mice ranged from ages 6 to 22 months. We also had a fairly even distribution of sex. We acquired our images using a 7 Tesla Bruker scanner with volume excitation coils and a 4-channel surface receiver array. We also acquired our resting state fMRI images using a T2 star echoplanar imaging protocol with around 300 volumes, and volumes are time points. We did this for 10 minutes to generate images with a 0.3 millimeter resolution. For anatomical scans, we used the rare protocol. We did a turbo rare and a regular rare. We did this for around 20 minutes, and we generated images with 0.1 millimeter resolution. After acquiring our images, we began the pre-processing pipeline. The first step of pre-processing is to correct for the bias field. This means that we correct for depth-dependent sensitivities from the 4-channel surface receiver within the scanner. We also do skull stripping to make sure that we only include the voxels that belong to the actual brain. The second step is to register our T1s to the same anatomical space. We also reorient them with the RAI orientation. And the last step is to resample our T1s with a 0.1 millimeter resolution and to port them with atlas labels. This helps us when we do our analysis because we want to make sure that specific regions align with the correct labels. After we pre-process our structural images, we pre-process the functional images. Again, like the structural images, we do a bias field correction and we perform skull stripping. The second part is to do a registration, which means we align our structural images with the anatomical image and the mouse brain atlas. Thirdly, we perform detrending and despiking on the bold signal. The bold signal is the blood oxygen level dependent signal. We also do confound correction, specifically within removing signal from white matter, cerebrospinal fluid, and blood. We use the blood oxygen level dependent signals to generate connectomes. Connectomes are the Pearson correlation between the bold time series of different regions. I have some examples of connectomes on the screen. We can see a connectome for younger mice and older mice, and hopefully you can see some very strong visible differences between these connectomes. They're averaged together. We feed all of our pre-processed images into a tool called FSL. The first part of our analysis is group-independent component analysis. This means that we represent our fMRI data as a mixture of several statistically independent signals. Group ICA assumes that we can take this data and separate it into 20 spatial maps and temporal maps. On the screen, I have 20 example spatial maps that we actually generated, and you can see a positive and a negative correlation in specific regions. Then we input our ICA components into the dual regression process. Dual regression applies a generalized linear model to our ICA components and our fMRI data. The first step of dual regression is spatial regression. This means that we compute subject-specific time courses by representing our group ICA components as regressors in our model. After that, we take these subject-specific temporal data, and we apply them as regressors in the second step, where we compute subject-specific spatial data. So it's this idea of taking all this group data and separating it into a subject level. The last step is permutation testing. This means that we do 5,000 permutations of our input data, and we also do threshold-free cluster enhancement to make sure that we have truly significant results. The first result that we produced from dual regression was the diet effect. On the left side, you can see some p-value corrected maps for the high-fat diet versus control diet effect. On the right side, I show the ICA components that had the biggest contributions to our results. When high-fat diet had a greater connectivity than control diet, we noticed much larger clusters, specifically within the piriform cortex and the frontal association cortex. You can also see other regions listed on the screen. But when control diet had a stronger connectivity than high-fat diet, we saw some much smaller areas, specifically in the primary somatosensory cortex and the hippocampus. We did another dual regression test within the Kahn toolbox, where we did the 50 oldest mice and the 50 youngest mice. So that was a sample size of 100 mice. On the left side, you can see regression for the age effect. We noticed some small clusters within the inferior colloquus, or the striatum. On the right side, I did a two-sample t-test where I compared younger mice and older mice. I noticed the olfactory area as a cluster, as well as the entorhinal cortex. After we did some more local analysis, specifically on the regions in the brain that we noticed specifically with our contrasts, we did topological analysis. This means that we represent our connectome, or the connectivity matrix that I showed earlier, as a graph. So brain regions are nodes, and edge weights are Pearson correlations. We compute the clustering coefficient, which is the degree to which nodes tend to cluster together. Here are some examples of clustering coefficients for genotype, age, sex, and diet. We noticed that E333 had a higher clustering coefficient, while younger mice had a clustering coefficient, as well as females, and lastly, the control diet. We did another test for path length, where we compute the number of edges it takes to traverse from one node to another node. So this is basically the inverse of network efficiency. We computed this again for the same types of groups. We noticed here that E44 had a higher path length, while older mice had a higher path length, males had a higher path length, and high-fat diet had a higher path length. There's actually mild differences between our analysis on the local and global level. This is a summary of our results. We noticed that high-fat diet resulted in lower clustering, but higher path length. When high-fat diet had stronger connectivity than the control diet, we saw specific regions like the insular cortex, the piriform cortex, but there are also much larger clusters. And this was not aligned with our initial hypothesis, so it was a bit of a surprising result. Our other contrast, which is when control diet had a stronger connectivity than high-fat diet, we saw some regions in the hippocampus and the cingulate cortex. For APOE4, we did more of a topological analysis where we noticed a lowering clustering coefficient and a higher path length. We also computed some statistics for age, like a regression and a young versus old. The higher the age, the lower the clustering coefficient, and the higher the path length. We noticed the hippocampus and motor cortex for the age effect, and we noticed olfactory areas when we were comparing younger mice to older mice. For sex, we were not able to get any significant results from dual regression, but we noticed on a global topological level that females had a higher clustering coefficient and a lower path length. For future work, we hope to implement other contrasts. We want to understand the interaction term, specifically sex or genotype interaction. We also want to look at the role of immunity. So in previous presentations, we saw that immunity actually plays a very strong effect on the risk for Alzheimer's disease. We hope to use our data on replacing the mouse NOS2 gene with the human NOS2 gene, and we want to investigate the effect of exercise. So that was just a general gist of my presentation, and I'm open to any questions that you might have, or even recommendations for future work.